1. Which part of the antibody molecule is responsible for binding to antigens? A Fab region. B. Fc region. C. Heavy chain. D. Light chain. Answer. A Fab region. 2019. 2. Which of the following antibodies is primarily found in mucosal surfaces? A. IgA. B. IgM. C. Ig. D. Ig. Answer. A. IgA. 2020. 3. What is the primary function of Ig antibodies in the immune response? A. Activation of complement system and neutralization of pathogens. B. Killing of infected cells. C. Prevention of allergic reactions. D. Production of antibodies by plasma cells. Answer. A. Activation of complement system and neutralization of pathogens. 2021. Conclusion. Antibodies are vital molecules in the adaptive immune system, responsible for recognizing, neutralizing, and eliminating pathogens through various mechanisms such as neutralization, opsonization, complement activation, and ADCC. Their structure, with distinct Fab and Fc regions, allows them to perform these diverse functions effectively. Understanding antibody structure and function is essential in immunology, vaccine development, and therapeutic interventions. You said. Generation of antibody diversity. ChatGPT said. Generation of antibody diversity. The immune system has a remarkable ability to generate an immense diversity of antibodies to recognize virtually any pathogen or foreign substance. This diversity is critical for the adaptive immune response, enabling the immune system to respond to a vast array of antigens. The generation of antibody diversity occurs primarily in B cells and involves several mechanisms, which include genetic rearrangement, somatic hypermutation, and class switching. 1. Genetic rearrangement (VTJ) recombination. The first mechanism responsible for antibody diversity is VTJ recombination, a process that occurs during the development of B cells in the bone marrow. This involves the rearrangement of specific gene segments in the immunoglobulin Ig loci, which encode the variable regions of the heavy and light chains of antibodies. A heavy chain locus. The heavy chain of the antibody is encoded by 3 gene segments, variable B diversity T and joining J. These segments undergo rearrangement in a process called VTJ recombination. B genes. There are multiple B gene segments in the heavy chain locus. D genes. There are several D gene segments in the heavy chain locus. J genes. A series of J gene segments are available for recombination. During VTJ recombination, 1B, 1D, and 1J segments are selected at random and recombined to form a complete gene that codes for the variable region of the heavy chain. This process introduces a high degree of variability, as the selection of different B, D, and J segments leads to the generation of diverse antibodies. B. Light chain locus. The light chains of antibodies are encoded by B and J gene segments, and they undergo a similar recombination process. However, unlike the heavy chain, there is no D segment in the light chain locus. Kappa kappa light chain, the most common light chain type in humans. The kappa chain locus has many B and J segments. Lambda lambda light chain, the second type of light chain, which also undergoes VJ recombination but has a different set of B and J segments compared to the kappa light chain. In B cells, recombination of the B and J gene segments occurs to form the variable region of the light chain. This recombination contributes to the overall diversity of the antibody repertoire. See mechanism of recombination. The recombination of B, D, and J segments is mediated by a group of enzymes, most notably RAG1 and RAG2 recombination activating genes. These enzymes introduce double-strand breaks in the DNA and facilitate the rearrangement process. The resulting recombined genes are transcribed, and the corresponding antibodies are translated. 2. Junctional diversity. Junctional diversity is another mechanism that enhances the diversity of the antigen binding site during VTJ recombination. The process occurs at the junctions where the B, D, and J segments are joined. Exonuclease activity. After the B, D, and J segments are brought together, exonucleases remove a few nucleotides at the joining sites, introducing addition. One. Genetic rearrangement VTJ recombination. The first mechanism responsible for antibody diversity is VTJ recombination, a process that occurs during the development of B cells in the bone marrow. This involves the rearrangement of specific gene segments in the immunoglobulin Ig loci, which encode the variable regions of the heavy and light chains of antibodies. A heavy chain locus. The heavy chain of the antibody is encoded by 3 gene segments, variable B diversity T and joining J. These segments undergo rearrangement in a process called VTJ recombination. B genes. There are multiple B gene segments in the heavy chain locus. D genes. There are several D gene segments in the heavy chain locus. J genes. A series of J gene segments are available for recombination. During VTJ recombination, 1B, 1D, and 1J segments are selected at random and recombined to form a complete gene that codes for the variable region of the heavy chain. This process introduces a high degree of variability, as the selection of different B, D, and J segments leads to the generation of diverse antibodies. B. Light chain locus. The light chains of antibodies are encoded by B and J gene segments, and they undergo a similar recombination process. However, unlike the heavy chain, there is no D segment in the light chain locus. Kappa kappa light chain, the most common light chain type in humans. The kappa chain locus has many B and J segments. Lambda lambda light chain, the second type of light chain, which also undergoes VJ recombination but has a different set of B and J segments compared to the kappa light chain. In B cells, recombination of the B and J gene segments occurs to form the variable region of the light chain. This recombination contributes to the overall diversity of the antibody repertoire. See mechanism of recombination. The recombination of B, D, and J segments is mediated by a group of enzymes, most notably RAG1 and RAG2 recombination activating genes. These enzymes introduce double-strand breaks in the DNA and facilitate the rearrangement process. The resulting recombined genes are transcribed, and the corresponding antibodies are translated. 2. Junctional diversity. Junctional diversity is another mechanism that enhances the diversity of the antigen binding site during VTJ recombination. The process occurs at the junctions where the B, D, and J segments are joined. Exonuclease activity. After the B, D, and J segments are brought together, exonucleases remove a few nucleotides at the joining sites, introducing additional variability. TTT terminal deoxynucleotidyltransferase. This enzyme adds non-templated nucleotides at the junctions of the B, D, and J segments. This process increases the variability of the antigen binding sites, contributing to the vast diversity of antibodies. The combination of the random selection of gene segments and the nucleotide addition at the junctions creates an immense diversity of combinations that are further enhanced by other mechanisms. 3. Somatic hypermutation. Once a B cell is activated by an antigen and undergoes clonal expansion, it can undergo somatic hypermutation in the germinal centers of lymphoid organs, such as lymph nodes and the spleen. This mechanism introduces point mutations into the variable region of the antibody genes, particularly in the B region. 8. Activation-induced cytidine deaminase. This enzyme is responsible for inducing mutations by deaminating cytosine residues in the DNA, leading to mutations that are repaired by error-prone repair mechanisms. Result of hypermutation. This leads to the generation of antibodies with increased affinity for the antigen, a process known as affinity maturation. B cells producing antibodies with higher affinity for the antigen are selected for survival, enhancing the effectiveness of the immune response over time. 4. Isotype class switching. In addition to generating diversity in the antigen binding site, B cells can also undergo class switching, or isotype switching, which changes the constant region of the antibody molecule while maintaining the same variable region. This process allows a single B cell to produce antibodies of different classes IgM, Ig, IgA, Ig that can carry out different immune functions. This process allows a single B cell to produce antibodies of different classes IgM, Ig, IgA, Ig that can carry out different immune functions, depending on the needs of the immune system. Class switching mechanism. Class switching is controlled by signals that activate the switch S regions in the immunoglobulin heavy chain locus. The activation-induced deaminase 8 enzyme plays a key role in initiating class switching. During this process, the DNA between the constant region genes is excised, allowing the antibody to be expressed as a different isotype. Effective class switching. The antigen specificity of the antibody remains unchanged, but the effector functions differ based on the isotype. For example, IgM antibodies are involved in complement activation, while the antibodies can facilitate opsonization and neutralization. 5. Alternative splicing of mRNA. In addition to the BTJ recombination, alternative splicing of the primary mRNA transcript also contributes to antibody diversity. A single B cell can produce both membrane-bound and secreted forms of antibody by alternative splicing. The membrane-bound form is used to anchor the antibody to the surface of the B cell, while the secreted form is released into the bloodstream to target pathogens. 6. Memory and antibody diversity. The ability to generate diverse antibodies is crucial for the development of immune memory. After an initial exposure to an antigen, some of the activated B cells differentiate into memory B cells. These memory cells have a long lifespan and can rapidly generate high-affinity antibodies upon re-exposure to the same pathogen. 7. Previous year questions and answers. 1. Which of the following processes is responsible for generating antibody diversity by combining different gene segments? A. Somatic hypermutation. B. BTJ recombination. C. Class switching. D. Junctional diversity. Answer. B. BTJ recombination. 2018. 2. Somatic hypermutation results in which of the following? A change in the antibody's isotype. B. Increase in the affinity of the antibody for the antigen. C. Rearrangement of the constant region genes. D. Generation of memory B cells. Answer. B. Increase in the affinity of the antibody for the antigen. 2019. 3. What is the role of A. Activation-induced cytidine deaminase in antibody diversity? A. It initiates BTJ recombination. B. It induces somatic hypermutation. C. It facilitates class switching. D. It enhances junctional diversity. Answer. B. It induces somatic hypermutation. 2020. Conclusion. The generation of antibody diversity is a crucial process that enables the immune system to recognize and combat a wide variety of pathogens. Through BTJ recombination, somatic hypermutation, and class switching, B cells produce antibodies with immense variability in their antigen binding sites, effector functions, and specificity. These mechanisms ensure the development of a highly adaptive and effective immune response capable of defending against an array of infectious agents. You said. Monoclonal antibodies. ChatGPT said. Monoclonal antibodies. Monoclonal antibodies (mAbs) are laboratory-produced molecules engineered to serve as substitutes for natural antibodies. They are made by creating identical copies, clones, of a single immune cell, which produces one type of antibody, hence the term monoclonal. These antibodies are used for a wide range of applications, including diagnosis, treatment of diseases, and research. The invention of monoclonal antibody technology was a landmark achievement in immunology and biomedicine. 1. Introduction to monoclonal antibodies. Monoclonal antibodies are highly specific and bind to only one epitope of an antigen. This specificity makes them invaluable in both therapeutic and diagnostic settings. Unlike polyclonal antibodies, which are derived from multiple B cell clones, monoclonal antibodies are produced by a single clone of B cells, allowing for high consistency and reproducibility. Monoclonal antibodies are used in the treatment of various cancers, autoimmune diseases, and infectious. Major histocompatibility complex (MHC) molecules. CSIR-Net Life Sciences Lecture Introduction and Basic Concept. Major histocompatibility complex (MHC) molecules are a group of highly polymorphic glycoproteins found on the surface of cells that play a pivotal role in the adaptive immune response. They are responsible for presenting peptide fragments derived from pathogens to T cells, thus enabling the immune system to distinguish between self and non-self antigens. The genes encoding MHC molecules are clustered in a genomic region known as the MHC locus, which is located on chromosome 6 in humans. This locus is called HLA, human leukocyte antigen complex in humans. Classes of MHC molecules. There are two primary classes of MHC molecules. Classes of MHC molecules. There are two primary classes of MHC molecules. 1. MHC class 1 molecules. MHC class 1 molecules are expressed. MHC class 1 molecules are expressed on all nucleated cells. They present endogenous antigens typically derived from intracellular pathogens like viruses to CD8+ cytotoxic T lymphocytes. Each MHC class 1 molecule is composed of a heavy alpha chain with alpha 1, alpha 2, and alpha 3 domains and a light chain called beta 2 microglobulin. The peptide binding groove is formed by the alpha 1 and alpha 2 domains. MHC class 1 molecules bind peptides of 8 to 10 amino acids in length. The expression of MHCI is critical for the immune surveillance of infected or transformed cells. 2. MHC class 2 molecules. MHC class 2 molecules are expressed primarily on professional antigen-presenting cells, APCs, such as dendritic cells, macrophages, and B cells. They present exogenous antigens originating from outside the cell to CD4+ helper T cells. Each MHC class 2 molecule consists of two transmembrane chains, alpha and beta, each with two domains alpha 1, alpha 2, beta 1, beta 2. The peptide binding groove is formed by the alpha 1 and beta 1 domains. They typically bind peptides of 13 to 25 amino acids in length. MHC2 molecules initiate immune responses against extracellular pathogens. Antigen processing and presentation pathways. MHC class 1 pathway. Intracellular proteins are degraded by the proteasome into peptides. These peptides are transported into the endoplasmic reticulum by TAP transporter associated with antigen processing proteins. Peptides bind to MHCI molecules in the ER. The complex is transported via the Golgi to the cell surface. The complex is transported via the Golgi to the cell surface. MHC class 2 pathway. Extracellular antigens are internalized via endocytosis or phagocytosis. These are degraded in acidic endosomal compartments. MHC2 molecules, synthesized in the ER, are stabilized by an invariant chain E that blocks the binding groove. In late endosomes, the invariant chain is degraded to CLIP class 2 associated invariant chain peptide. HLA-DM facilitates the removal of CLIP and loading of antigenic peptides. The MHC2 peptide complex is expressed on the cell surface. Polymorphism and genetic organization. MHC genes are highly polymorphic, meaning there are many different alleles in the population, allowing for diverse antigen presentation. This polymorphism is concentrated in the peptide binding regions, contributing to the breadth of immune responses. MHC genes are inherited codominantly, so both maternal and paternal alleles are expressed. The HLA gene complex includes Class 1 genes, HLA-A, HLA-B, HLA-C. Class 2 genes, HLA-DP, HLA-DQ, HLA-drive. Class 3 region contains genes for complement proteins C2, C4, factor B, and cytokines like TNF. MHC restriction and T cell recognition. T cells recognize antigens only when presented by self-MHC molecules, a phenomenon known as MHC restriction. CD8+ T cells recognize antigen only in the context of MHCI, while CD4+ T cells recognize antigens in the context of MHC2. This ensures specificity and proper coordination of immune responses. Clinical relevance. Transplantation. MHC matching is crucial for graft acceptance. Mismatched MHC leads to graft rejection. Autoimmune diseases. Certain HLA alleles are associated with increased risk, e.g., HLA-B27 with ankylosing spondylitis. Infectious diseases and vaccines. MHC variability affects individual responses to pathogens and vaccines. Previous year CSIR-NET questions. 1. CSIR-NET Deck 2014. Which of the following cells is most likely to express MHC class II molecules? A. Red blood cells. B. Neurons. C. B cells. D. Skeletal muscle cells. Answer. C. B cells. 2. CSIR-NET June 2016. The binding group of MHC class I molecule is formed by which domains? A. Alpha 1 beta 1. B. Alpha 2 and beta 2. C. Alpha 1 and alpha 2. D. Alpha 3 and beta 2 microglobulin. Answer. C. Alpha 1 and alpha 2. 3. CSIR-NET Deck 2017. Which molecule facilitates the exchange of clip with antigenic peptide on MHC class II? A. TAP. B. Calnixin. CHLADM. D. Topicin. Answer. CHLADM. Would you like the next lecture on T-cell activation and co-stimulatory molecules or antigen processing in detail with diagram-based explanations? You said. Antigen processing. ChatGPT said. Here's a CSIR-NET JRF Life Sciences level. This will serve as perfect study material for your exam preparation. Antigen processing. CSIR-NET Life Sciences lecture introduction. Antigen processing is the intracellular mechanism by which antigenic proteins are broken down into peptide fragments suitable for presentation on the cell surface by MHC molecules. This process is a critical component of the adaptive immune response because T lymphocytes do not recognize native, intact antigens but rather recognize short peptides bound to MHC molecules. The system ensures that both intracellular and extracellular pathogens are presented to the appropriate subsets of T cells, cytotoxic T cells CD8+, and helper T cells CD4+, respectively. Antigen processing is tightly linked with antigen presentation and is executed via two main pathways: the cytosolic endogenous pathway associated with MHC class I, and the endocytic exogenous pathway associated with MC class II. A third, less common route is the cross-presentation pathway, where exogenous antigens are presented on MHCI molecules, particularly in dendritic cells. 1. Cytosolic endogenous pathway, MHC class I presentation. This pathway processes antigens derived from intracellular sources, such as viral proteins, tumor antigens, or any protein synthesized within the cytosol of a cell. The process begins with the ubiquitination of defective or aged proteins, targeting them for degradation by the 26S proteasome, a multicatalytic protease complex. The resultant peptide fragments are typically 8 to 16 amino acids long. These peptides are then transported into the endoplasmic reticulum by a specialized transporter called TAP, transporter associated with antigen processing, which is an ATP-dependent heterodimer composed of TAP1 and TAP2. Inside the the peptides are loaded onto newly synthesized MHC class I molecules, which are stabilized by a complex of chaperone proteins including calnixin, calreticulin, ERK57, and topicin. Topicin bridges the MHC class I molecule to TAP, ensuring high-affinity peptide loading. Once the peptide MHCI complex is stabilized, it is transported through the Golgi apparatus to the plasma membrane, where it presents the antigenic peptide to CD8+ cytotoxic T lymphocytes. This leads to the killing of the infected or transformed cell. 2. Endocytic exogenous pathway, MHC class II presentation. The exogenous pathway is specialized for processing antigens that originate outside the cell, such as bacterial toxins, extracellular viruses, or soluble proteins. These antigens are taken up into the cell via phagocytosis, receptor-mediated endocytosis, or pinocytosis, and are directed into acidified endosomal or lysosomal compartments. Inside these compartments, the proteins are degraded by acid proteases like cathepsins into peptides that are typically 13 to 25 amino acids in length. Meanwhile, MHC class II molecules are synthesized in the where they associate with an invariant chain P. This chain stabilizes the MHC molecule and blocks premature peptide binding. The MHC2 invariant chain complex is transported to MIIC compartments, MHC class II compartments via the Golgi apparatus. In these late endosomal compartments, the invariant chain is degraded, leaving a residual peptide fragment called CLIP, class II associated invariant chain peptide, in the binding groove. The molecule HLADM then catalyzes the removal of CLIP and facilitates the loading of antigenic peptides onto the MHC class II molecule. The final peptide MHC class II complex is transported to the cell surface, where it is recognized by CD4+ helper T cells, which then initiate a broad immune response including the activation of B cells, macrophages, and cytotoxic T cells. Cross-presentation and cross-priming. Some specialized antigen-presenting cells, especially dendritic cells, can process exogenous antigens and present them on MHC class I molecules, a phenomenon called cross-presentation. This allows the immune system to generate CD8+ cytotoxic T cell responses against viruses or tumor antigens that are not directly infecting APCs. Cross-priming refers to the activation of naive CD8+ T cells via cross-presentation. Cross-presentation is crucial in cancer immunity and viral infections where infected non-APCs need to be recognized by CD8+ T cells. The mechanisms behind cross-presentation include the cytosolic pathway, where internalized antigens escape the endosome and enter the cytosol for proteasomal degradation, and the vacuolar pathway, where peptides are generated within endosomes without involving the proteasome or TAP. 4. Regulation and immunological importance. The efficiency and fidelity of antigen processing are tightly regulated, as errors can lead to immune evasion by pathogens or the development of autoimmune diseases. For instance, mutations in TAP genes can lead to bare lymphocyte syndrome, a condition with severely reduced MHCI expression. Similarly, defects in invariant chain or HLA-DM can impair MHC2-mediated presentation and compromise helper T cell responses. MHC polymorphism also plays a role in determining the spectrum of peptides presented, influencing susceptibility or resistance to infections, autoimmune diseases, and vaccine responsiveness. Previous year CSIR-NET questions. 1. CSIR-NET June 2015. The transporter that helps in loading peptides into the for MHC class I presentation is a HLA-DM. Clip circuit TAP. D invariant chain answer, C-TAP. 2. CSIR-NET Deck 2017. What is the role of the invariant chain in MHC class II processing? A degrades antigen. B facilitates TAP binding. C prevents premature peptide binding. D inhibits T cell activation. Answer, C prevents premature peptide binding. 3. CSIR-NET June 2019. Which of the false statements is true regarding cross-presentation? A. It is exclusive to B cells. B. It presents endogenous antigen on MHC2. C. It allows extracellular antigen to be presented on MHCI. D. It requires invariant chain. Answer, C. It allows extracellular antigens to be presented on MHCI. You like the next lecture on T cell activation and signaling or a deep dive into cross-presentation and viral immune evasion mechanisms? You said. Antigen presentation, ChatGPT said. Here is your CSIR-NET JRF Life Sciences level lecture on antigen presentation, crafted in a paragraph format with maximum detail, based on previous year questions, with no tables, and including 2 to 3 solved CSIR questions with years. This note is designed to be your perfect and only study material before the exam. Antigen presentation, CSIR-NET Life Sciences lecture introduction. Antigen presentation is a vital immunological process in which cells display processed antigenic peptides on their surface in association with major histocompatibility complex (MHC) molecules to be recognized by T cells. It forms the essential bridge between innate and adaptive immunity, ensuring that T lymphocytes are exposed to peptides derived from both intracellular endogenous and extracellular exogenous sources. Without antigen presentation, T cells cannot recognize and respond to pathogens, tumors, or even self-antigens, which makes this process central to immunological surveillance, defense, and tolerance. MHC molecules and their role in antigen presentation. There are two main classes of MHC molecules involved in antigen presentation, MHC class 1 and MHC class 2, each with distinct structural features, peptide sources, expression patterns, and T cell targets. MHC class 1 molecules are expressed on all nucleated cells and present endogenous antigens, typically derived from intracellular pathogens like viruses or altered self proteins such as tumor antigens. These processed peptides, generated by the proteasome and transported into the endoplasmic reticulum by TAP proteins, are loaded onto MHCI molecules and presented to CD8+ cytotoxic T lymphocytes, CTLs. This leads to the targeted destruction of infected or transformed cells. In contrast, MHC class 2 molecules are expressed primarily on professional antigen-presenting cells, APCs, such as dendritic cells, macrophages, and B cells. These molecules present exogenous antigens, such as bacterial proteins or toxins, which have been internalized via endocytosis or phagocytosis and processed in acidified endosomal compartments. The resulting peptides are loaded onto MHC2 molecules after the removal of the invariant chain and clipped by HLA-DM. These MHC2 peptide complexes are then presented to CD4+ helper T cells, which orchestrate broader immune responses including B cell activation and cytokine secretion. Mechanism of antigen presentation. Antigen presentation is tightly coupled with antigen processing, where the antigenic proteins are broken down into smaller peptides suitable for loading onto MHC molecules. The MHC peptide complex must be stably formed to be transported to the cell surface for effective recognition by T cells. For MHCI presentation, the antigenic peptides are typically 8 to 10 amino acids long and must fit precisely into the binding groove. For MHC2, the peptides are 13 to 25 amino acids and can bulge outside the groove due to the open-ended nature of the peptide binding cleft. This difference in structure directly influences the nature and diversity of peptides presented by each MHC class. Upon presentation, CD8+ T cells recognize the MHCI peptide complex and become activated to release perforin and granzymes, inducing apoptosis in the target cell. CD4+ T cells, recognizing MHC2 peptide complexes, differentiate into subsets like Th1, Th2, Th17, or Treg cells, each performing specialized immune functions. Importantly, T cell recognition is MHC restricted, meaning that the T cell receptor TCR recognizes both the peptide and the MHC molecule simultaneously. Specialized antigen presentation pathways. A unique form of presentation known as cross-presentation allows certain APCs, especially dendritic cells, to present exogenous antigens on MHCI molecules, thereby activating CD8+ T cells. This is essential for immunity against viruses or tumors that do not infect APCs directly. Cross-priming refers to the activation of naive CD8+ T cells via this pathway. Another specialized mechanism is autophagy-mediated presentation, where intracellular components are degraded and presented on MHC2 molecules, important in immune surveillance and tolerance. Immunological significance. Antigen presentation is critical for distinguishing self from non-self and activating appropriate immune responses. A failure in antigen presentation, such as mutations in TAP genes, HLA-DM, or MHC genes, can lead to immune deficiencies, autoimmune diseases, or immune evasion by pathogens. Many viruses, including herpes viruses and cytomegalovirus, have evolved strategies to interfere with MHC expression or antigen processing to escape immune detection. Additionally, antigen presentation plays a central role in vaccine development, where epitopes must be selected based on their ability to bind to a broad range of MHC alleles in the population. The diversity of MHC alleles, high polymorphism, ensures that different individuals present different sets of peptides, influencing disease susceptibility, autoimmunity, and transplant rejection. CSIR-NET previous year questions. 1. CSIR-NET June 2016. Which of the following cells are capable of presenting antigens via MHC class 2 molecules? A. Red blood cells. B. Epithelial cells. C. Dendritic cells. D. Neurons. Answer. C. Dendritic cells. 2. CSIR-NET Deck 2018. Which of the following molecules is directly involved in stabilizing peptide binding to MHC class 2 by replacing CLIP? A. Topicin. B. HERP57 CHLA-DM. D. Calreticulin. Answer. CHLA-DM. 3. CSIR-NET June 2021. Cross-presentation is a process where endogenous antigens are presented on MHC2. B. Exogenous antigens are presented on MHCI. C. Endogenous antigens are presented on MHCI. D. MHC2 molecules present like lipids. Answer. B. Exogenous antigens are presented on MHCI. Conclusion. Antigen presentation is a cornerstone of adaptive immunity that ensures T cells are accurately activated in response to specific antigenic peptides presented by MHC molecules. The efficiency, specificity, and fidelity of this process are essential not only for defending against infections and tumors but also for maintaining immune homeostasis. Mastery of this topic is vital for CSIR-NET, as it is frequently targeted in conceptual and application-based questions. Would you like the next lecture on T cell activation and signaling pathways, or should we move on to B cell receptor signaling and antibody production? You said, activation and differentiation of B cells. ChatGPT said. Here is your CSIR-NET JRF Life Sciences level lecture on activation and differentiation of B cells, written in detailed, paragraph form with maximum word limit, no tables, and 2 to 3 CSIR-NET previous year questions with answers and years mentioned. This lecture is fully exam-oriented and structured to serve as your only and perfect study material. Activation and differentiation of B cells, CSIR-NET Life Sciences lecture introduction. B lymphocytes. B cells are a vital component of the adaptive immune system, responsible for the production of antibodies and the establishment of humoral immunity. The activation and differentiation of B cells transform them from naive, antigen-inexperienced cells into antibody-secreting plasma cells and long-lived memory B cells. This process involves a sequence of highly regulated events initiated by antigen recognition, further amplified by signals from helper T cells, cytokines, and co-stimulatory molecules. The end result is the production of high-affinity, isotype-switched antibodies and immunological memory, cornerstones of effective immunity and vaccine response. Naive B cells and antigen encounter. Naive B cells originate from the bone marrow and migrate to secondary lymphoid organs, such as the lymph nodes and spleen, where they reside in follicles. These cells express membrane-bound immunoglobulin, made primarily IgM and IgD, which function as the B cell receptor BCR complex. The BCR complex consists of the surface immunoglobulin molecule associated with signaling proteins Ig-alpha, CD79, and Ig-beta, CD79b, which transmit intracellular signals upon antigen engagement. The first signal in B cell activation is provided when the BCR binds a specific antigen with high affinity. This binding triggers receptor cross-linking and clustering, leading to phosphorylation of ITAMs on Ig-alpha and Ig-beta by SRC family kinases, e.g., Lin recruitment of SYK kinase, and activation of downstream pathways such as MEPK, PI3K, and NF-kappa B, leading to changes in gene expression. However, this signal alone is not sufficient for full activation in most cases. T-dependent B cell activation. Most protein antigens require T cell help and are termed T-dependent T-antigens. Once BCRs bind the antigen, the complex is internalized, processed, and the resulting peptide fragments are presented on MHC class II molecules to CD4 plus helper T cells. The interaction between the MHC2 peptide complex on B cells and the T cell receptor TCR on T helper cells forms the basis of the second signal. Additional co-stimulatory interactions are critical for full B cell activation. The most crucial of these is the CD40-CD40L CD154 interaction, where CD40 on B cells binds CD40L expressed on activated helper T cells. This binding triggers a cascade involving NF-kappa B and AP1 that promotes B cell proliferation, survival, isotype switching, and somatic hypermutation. T helper cells also secrete cytokines like IL-4, IL-5, IL-6, and IL-21, which modulate the direction and magnitude of B cell responses. Germinal center reaction and B cell differentiation. Activated B cells either differentiate rapidly into short-lived plasma cells in extrafollicular regions or migrate into follicles to form germinal centers, specialized microenvironments where they undergo clonal expansion, somatic hypermutation (SHM), and class switch recombination (CSR). Somatic hypermutation introduces point mutations into the variable regions of the immunoglobulin genes, leading to a spectrum of BCR affinities. Affinity maturation is driven by the selection of B cells that bind antigens with higher affinity, with the help of follicular dendritic cells (FDCs) and T follicular helper (TFH) cells. B cells expressing higher affinity receptors receive survival signals and outcompete others. Class switch recombination involves changing the constant region of the heavy chain, e.g., from IgM to Ig, IgA, or Ig without altering antigen specificity. This process, guided by activation-induced cytidine deaminase, is influenced by cytokines. IL-4 promotes Ig, IFN-gamma promotes Ig2A, TGF-beta promotes IgA, etc. The germinal center response culminates in the differentiation of B cells into long-lived plasma cells that secrete high-affinity antibodies or into memory B cells, which persist for years and respond rapidly upon re-exposure to the antigen. T-independent B cell activation. Some antigens, such as bacterial polysaccharides or lipopolysaccharides, can activate B cells without T cell help and are classified as T-independent TI antigens. These are divided into type 1, e.g., LPS, which act as polyclonal B cell activators, and type 2, e.g., repetitive polysaccharides, which extensively cross-link BCRs. TI responses lead to rapid production of IgM antibodies but usually do not generate memory B cells or involve affinity maturation or class switching, making them less effective in long-term protection. Terminal differentiation. Following activation, B cells differentiate into plasma cells, terminally differentiated, antibody-secreting cells that localize to the bone marrow or mucosal site. They have abundant rough endoplasmic reticulum for antibody production and lose BCR expression. Memory B cells, long-lived, antigen-experienced cells that circulate or reside in secondary lymphoid tissues. They respond rapidly and robustly upon secondary exposure, contributing to long-lasting immunity. Regulation and tolerance. Activation of B cells is tightly regulated to prevent autoimmunity. Central tolerance eliminates autoreactive B cells in the bone marrow via receptor editing or deletion. Peripheral tolerance mechanisms include energy, ignorance, and regulatory B cells (Bregs) that secrete IL tend to suppress inflammation. Dysregulation of B cell activation contributes to autoimmune disorders like systemic lupus erythematosus (SLE) and rheumatoid arthritis. CSIR-NET previous year questions. 1. CSIR-NET Deck 2017. Which molecule expressed by activated T cells is essential for isotype switching in B cells? AIL2. BCD40-LCB7D Facil. Answer: BCD40L. 2. CSIR-NET June 2019. Which enzyme is involved in both somatic hypermutation and class switch recombination? ARAG1. BTDTCA-DDNAPK. Answer: C8. 3. CSIR-NET Deck 2022. T-independent antigens typically induce which of the following responses? A strong Ig production. B high affinity antibody generation. C memory cell formation. D predominant IgM production. Answer: D predominant IgM production. Conclusion. The activation and differentiation of B cells are central to the adaptive immune response and critical for lifelong immunity. Through a sophisticated interplay of signals from the antigen, helper T cells, and cytokines, B cells undergo clonal expansion, somatic mutation, and class switching to produce potent antibodies and memory cells. This dynamic process ensures both immediate defense and long-term protection, making it an essential topic for CSIR-NET life sciences. Understanding the molecular mechanisms and regulatory controls governing B cell responses is key to interpreting vaccine function, immunodeficiency, and autoimmune pathologies. Would you like to continue with T cell activation and differentiation, or proceed to antibody structure and function? Said, activation and differentiation of T cells. ChatGPT said. Here is your CSIR-NET JRF Life Sciences level lecture on activation and differentiation of T cells, written in maximum detail paragraph format, with no tables, including 2 to 3 CSIR-NET previous year questions with answers and years mentioned. This content is tailored to serve as your only study material and coaching before the exam. Activation and differentiation of T cells, CSIR-NET Life Sciences lecture introduction. T lymphocytes. T cells are key effectors of the adaptive immune system and play pivotal roles in cellular immunity. T cells originate in the bone marrow but mature in the thymus, where they undergo positive and negative selection to ensure self-tolerance and functional competency. Once matured and released into circulation as naive T cells, they remain inactive until they encounter a specific antigen. The process of T cell activation and differentiation transforms these naive cells into functionally active effector T cells or long-lived memory T cells. This transformation is essential for orchestrating effective immune responses, including cytotoxicity, cytokine secretion, and regulation of immune homeostasis. Naive T cell activation. Signal 1. Antigen recognition. Naive T cells express a unique T cell receptor (TCR) on their surface, which is specific to a particular antigenic peptide presented on major histocompatibility complex (MHC) molecules by antigen-presenting cells (APCs). CD8 plus T cells recognize peptides presented by MHC class I, while CD4 plus T cells recognize those presented by MHC class II. The TCR complex comprises the TCR alpha and beta chains responsible for antigen recognition, along with the CD3 complex, which transduces intracellular signals through immunoreceptor tyrosine-based activation motifs (ITAMs). Signal 1 is initiated when the TCR binds to the peptide MHC complex, stabilized by CD4 or CD8 co-receptors depending on the T cell subtype. However, signal 1 alone is insufficient for full activation. Without additional signals, the T cell may become anergic, non-responsive, or undergo apoptosis. Signal 2. Company stimulation. The second signal, or co-stimulatory signal, is essential for complete activation and survival of T cells. The most prominent co-stimulatory interaction involves the binding of CD28 on the T cell with B7-1 CD80 or B7-2 CD86 molecules on the APC. This signal triggers downstream signaling cascades that activate transcription factors such as NF-kappa B, NF-AT, and AP1, leading to the production of interleukin-2 IL-2, a key growth factor for T cells. If TCR engagement occurs in the absence of co-stimulation, T cells enter a state of anergy, which is a peripheral tolerance mechanism to prevent autoimmunity. Signal 3. Cytokine environment and differentiation. The third signal is provided by cytokines in the microenvironment, which determine the direction of T cell differentiation. These cytokines are secreted by APCs or other immune cells in response to pathogen-associated molecular patterns (PAMPs). Upon receiving all three signals, naive T cells undergo clonal expansion and differentiate into various effector or memory subsets. This differentiation is driven by signal transduction pathways and transcription factors specific to the cytokine milieu encountered during activation. CD4 plus T cell differentiation. CD4 plus T cells differentiate into various helper T cell subsets, each characterized by specific cytokine profiles and functions. Th1 cells, induced by IL-12 and IFN gamma, regulated by the transcription factor TBET. They secrete IFN gamma and promote cell-mediated immunity, especially effective against intracellular pathogens. Th2 cells, induced by IL-4, governed by GATA3. They produce IL-4, IL-5, and IL-13, which activate eosinophils and B cells, important in helminth infections and allergic response. They secrete IL-17 and IL-22, enhancing neutrophil-mediated inflammation and mucosal defense. TFH follicular helper T cells, induced by IL-6 and IL-21, controlled by BCL6. They migrate to germinal centers and help B cells undergo affinity maturation and class switching. TREG regulatory T cells developed in the presence of TGF-beta, expressing FOXP3. They produce IL-10 and TGF-beta, suppressing excessive immune responses and maintaining self-tolerance. CD8 plus T cell differentiation. CD8 plus T cells differentiate into cytotoxic T lymphocytes, CTLs, which are crucial for killing virus-infected or tumor cells. Activation of CD8 plus T cells typically requires strong co-stimulation and often help from CD4 plus T cells. Once activated, CTLs release perforin and granzymes, which induce apoptosis in target cells. They can also trigger cell death via phosphactyl interactions. CTLs express surface markers like CD107A, granzyme B, and IFN gamma, and they circulate through tissues to survey and eliminate infected or transformed cells.